All right, this is chapter 6, part 3 of 3. So, last part of chapter 6, we're talking about how do we identify viruses, how do we get them, isolate them, and how do we culture them to study. So, isolating them is pretty simple. We just use a filter, and these filters have to have pores that are super, super small. So, remember that bacteria are about 100 to 1,000 times bigger than viruses. So these pores have to be small enough to let viruses through and then not let that bacteria through. So it's basically a membrane filter. Oops. A membrane filter that's used to filter out virus particles. So, oh, sorry, trying to get a pen going. So the size of the pores is going to determine like what's let through. And so basically you can just create these filters at any size. This is going to be a 5 micrometer pore. This is a 0.2 micrometer pore. So you can see that the bigger the size, you're going to get more objects being filtered. The smaller the size, the more fine and more exclusion you're going to have of larger things. So literally just, crushing something up, filtering it for viruses. And then how do we culture them? How do we grow viruses in a lab to study? Can we culture them in a broth or in a slant that we use in class or a petri dish? No, we can't because they require a host cell, right? They don't grow or reproduce unless they are infected, unless they have infected a cell. And so in order to grow viruses, we need cells for them to grow in. So we culture viruses to identify them, to isolate them for diagnosis. If it's a new virus, right, like COVID was, we really need to identify the virus and study it. Then we need to prepare if we want to make a vaccine. We culture viruses to make vaccines from. And just plain old straight up research, research what the virus does, how it infects the cell, etc. And then how, right? So there are these 3 ways. You can use tissue culture or cell culture. So these basically you have cells that are grown in a petri dish and you just infect the dish with virus and the virus will enter the cells. You can use bird embryos. In another word, eggs. Eggs are perfect. They're perfect for the viruses that infect birds. And the flu is one of those viruses. So all of our flu viruses are grown inside of eggs. Eggs are great because they're protected, they're sterile, they're in a self-sustaining environment, perfect for viral growth, right? So chicken, duck, turkey eggs are most used, and the egg is injected through the shell with the virus. Another way is through just live animals. So we can inject rats or mice or hamsters, all these animals with the virus. And even though that sounds really sad, it's what occurs to grow virus and study virus. This is an example of bacteriophages. Okay, so we have bacteria growing on the agar. So all the kind of yellowish, greenish background is going to be the bacterial culture. So bacterial cells are here. And then all of the open areas, right, they look blank, these little dots. Those clear areas are called plaques. So they're called viral plaques because in those areas, the virus has infected the bacterial cell. And remember, the bacterial cells always kind of lice, they die. And so they leave these little circles of emptiness because all the bacteria are dead here. And we can find virus here, right? So we can find our viruses in these plaques. So the term for those empty areas are called plaques, viral plaques. And the way that bacteria is growing here, it's not in colonies, it's just spread really evenly over the agar, so it doesn't form colonies. That's called a lawn of bacteria. So this is just another picture from your book on the actual image. So there are these little bottles that grow cells, and the red medium is the nutrition for those cells. It's the glucose, it's the oxygen, all the different things that the cells need to grow are in that red liquid. And then you can also just pop in some virus, so you know that the cells are going to get infected with the virus. And then this is just an example of some plates with E. coli grown on them. So they look milky white, milky yellow because of the bacteria. And then those plaques are where the bacteriophage has lysed the cells. Okay. So the viral titers increase on the plates to the left, right? So we can see that there's not much virus in this one, but as we go this way, the virus increases in number. Here's a picture of the eggs, right? So we have different areas depending on what virus you're working with, depending on different factors. You can actually inject the virus into different areas of an egg because an egg contains different tissues. It's not just one certain kind of cell. There's different kinds of cells in there. As you can see, there's all different kinds of structures inside of an egg. And so, yeah, they can be replicated in these guys. You can see that they're injecting these eggs with virus here. And some viruses are extremely dangerous. We still need to grow them and work with them so that we can study them, study how we can kill them, study their growth cycles. So what molecules they have on the surface. So this is something called, what this person is working in is a biosafety level 4 lab. So this is Ebola. Ebola is a virus that is really deadly. I think it has a mortality rate of 50%, so it's very, very virulent. These flasks all contain the Ebola virus growing in tissues, and this person is completely cut off from the air supply in the lab. So there is a suit that has its own air supply, and you can see those hoses here that you're going to attach to your suit so that you get an air supply. And you have to shower before, like you have to change clothes. It's a very large process to get in and work. But that's what the biosafety level 4 workers look like. Okay. This is an interesting cell line, and I want you guys to read that little area on HeLa cells. And maybe you've heard of HeLa. There is a movie about her. I think Oprah produced it. Henrietta Lacks was a Black woman who had cervical cancer, and they took some of her cells from her without her knowing, and they're using those cells, her cells, to grow virus. And her cells were so good at growing that they're still her cells today used to grow viruses. So they're called HeLa cells because HE stands for Henrietta, and this is Lax. So these HeLa cells are still around today. They were originally from her cervix, and some people like to use the word immortal because these cells have just, they're the same cell. It's, when cells divide in this manner, it's a clone, right? So mitosis, you make clones. And so we still have her cells alive today. So those cells are technically immortal. Like, I don't know if you agree with that, but you have the same genetic cell that was around in Henrietta Lacks as you do today. So do they ever die? If we keep them alive, will they ever die? So that's where the immortal comes from. So let's talk about this picture. So individual cells are isolated from tissue. So if you want to make a tissue culture, you're going to take some cells, put it in a petri dish, and normal cells have contact inhibition, meaning that when they grow into one layer on a petri plate and they touch each other, they stop growing. They do not grow in a mound. So they just have one layer of cells and that's it. So for culturing virus, this may be fine, but it's better to have more cells, right? So some of these cells are transferred to a new medium and you get a secondary culture. And so you can just keep the line going like this, right? You can keep making more plates of cells. In transformed cells, so the word transformed here means they're cancerous. Okay, so this is a cancerous cell, and they are from a tumor. And so in Henrietta Lacks's, right, it was from her cervix. And then you have this line, and you can see how the drawing is slightly different. We can get a mound of cells because tumor cells don't know when to stop. They lose their contact inhibition. They lose their ability to stop mitosis, the cycle. And so they just grow and grow and grow and grow into like this heap of cells. So you can see in letter B here, continuous cell cultures are not affected by contact inhibition. They continue to grow regardless of cell density. And so these are just two different ways that are used to make cell cultures for growing viruses or anything else really. But one, you have to be very careful. If it's a non-cancerous cell, you have to make sure you're always taking some cells and making another culture, because when they get crowded like that, they don't really like it. You know, you're competing for different things, making a lot of waste in here. So the waste will eventually build up. So these are, you know, really logistically just harder than it is to take care of cancerous or transformed cells. And this is our HeLa cells. They're just, you know, they use fluorescent microscopy on here. So you can see what's stained with what. All right, viruses cannot be cultured in. All right, and the answer is going to be culture media. So media refers to broth and agar, or yeah, that's it. So media, you can grow bacteria because they're cells and they can just like eat the different nutrients on the media, but viruses need a cell. So we need an animal, or we need a cell culture, or we need eggs. Okay, when a virus infects a cell, there are some visual or changes in the morphology of the cell. So I wanted to show you that then there is this term called syncytium. So that's kind of a tricky word to say. Syncytium. I just made a, now I can't say it anymore. Anyways, syncytiums are a fusion of several cells. So when one cell gets infected with virus, as you can see here, this is a virus particle. This is our cell that's infected. It will fuse with other cells, and then you get this really big cell with many nuclei, and it's making virus. So that's a syncytium. And so you can see this under a microscope as a cytopathic effect. So cyto is for cell, pathic is for disease, right? So this is signs of a diseased viral infection. There are other things you can see as well. So some viruses will make inclusion bodies. We looked at the word inclusion, I think it's in chapter 2, but inclusions are little areas inside a cell that store things. And these are not the greatest pictures, but you know, so then you're going to get little dots that show up inside the cells that show you that that's where the virus is. So the viruses are being stored in those little inclusions. Then you have cytoplasmic stranding. So some cells just take on this weird process, you know, a really long piece of their cytoplasm is formed. And you have nuclear inclusion bodies. And then here we have a grape-like cluster. So in these different examples, you can see that viruses, when they infect animal cells, they can lead to really weird changes of their shape, their morphology. How do we test for viruses? So this is one. There's a couple, I think, written in your book, but we're only going to talk about the hemagglutination. So COVID, when COVID first came out, they would do this hemagglutination. So we have red blood cells here. Let's see how we're going to talk about this. So hemagglutination, the heme stands for red blood cells, and agglutination is the clumping or gathering of the red blood cells. So you see here that if you have red blood cells and more red blood cells, there's no interaction and there's no reaction. In letter B, we have a virus with our red blood cells, and the virus can stick to more than one red blood cell. So normally red blood cells don't stick to each other. But when you have a virus that is attracted to the surface of red blood cells, you can actually make this really clumpy mass of red blood cells and virus, which is going to show up invisible to the eye as like little dots or areas of flecks of substance in the blood. In this image, we have virus and antibody and red blood cells. So instead of just clumping those red blood cells, what we have here is our body has made an antibody. This is going to be made by our bodies in response to a previous viral infection. So we only get antibodies when we've had the infection before. So in letter C, we have this protein called an antibody, and their job is to bind to the virus. You can see how that the antibody has bound to the virus, and this makes the virus unable to attach to our cells. So it makes it impossible for the virus to enter the cells and cause a new infection. And it also prevents those viral particles from agglutination. So it's not possible for them to clump these red blood cells. So when you see, if you put these together in a little test, we do not have hemagglutination. So when you have hemagglutination, this means that you have the virus in your body, and it's probably the first time you've been exposed to that because you don't have don't have antibodies yet. In letter C, the reaction is no hemagglutination inhibition, so no reaction, but it's because you've been infected before and you've made antibodies that are now counteracting the clumping mechanism of the virus. So you can tell by the reaction whether or not you have been exposed to the virus before because you have antibodies, or that you have an active infection and it's the first time because you have viruses clumping your red blood cells. Okay. All right, we're wrapping up with a different kind of infectious, I guess it's a protein, but infectious particle called a prion or prion. It stands for proteinaceous infectious particle, and it's literally just a protein. It's infectious protein. And it was pretty recently described as an infectious agent, and it's found in some animals, in sheep, in cows. It's also found in humans. So there are some genetic diseases that cause this prion to misfunction. So let me just say that the prion or prion that is mentioned here is a normal part of our nervous system. It's found on neurons in our brain, and it usually, doesn't cause any issue. However, sometimes people are born with the tendency of those proteins to become misfolded. And so when they become misfolded, those proteins then become very sticky and they like to stick to each other. And then when they stick to each other, they form this really big lump, right? So you have a lot of proteins forming a big clump, and this can be in your brain. And that is going to alert your immune system, and the immune system is going to come and kill. No immune system. The clumping is going to kill those neurons. So what happens in the genetic diseases is that there's something called fatal familial insomnia, where basically it's super, super rare. I think there's only one family in Italy that has been documented to have it, but they just can't go to sleep. One day they just can't go to sleep, and they can't go to sleep for the rest of their lives, and they will end up dying. And then Creutzfeldt-Jakob disease is another neurological disease that's caused by this, and it's also neurodegenerate. So your nervous system starts to just take a dive, and then death is 100%. Then there are the transmissible diseases. So this is the scary part, right? So that means you can get this disease from eating the bad protein shape. The diseases here are called bovine spongiform encephalopathy. This is otherwise known as mad cow disease. This is in cows. So cows have mad cows, which is called bovine, right? The word bovine stands for cow. Spongiform is what the brains of these cows look like after they've had this prion disease because of those holes. Remember, I talked about how your brain, when you have lots of those misfolded proteins, the cells will die. And so you're left with these holes inside of the brain. So it looks like a sponge. And encephalopathy, encepha is referring to the brain. Pathy is a disease. But notice it's not encephalitis. There's also a term encephalitis, and that's for the swelling of, different things. If you have itis, it's just a swelling or edema, right, of that area. But this doesn't cause swelling. So that's why I said, no, not the immune system. Bovine spongiform encephalopathy, there's no like big symptom of it that you have it because it doesn't cause that kind of immune response. So what it does is it just gradually degrades your nervous system. So that's a mad cow. So if a person were to eat some of this misshapen protein from a cow, then the human would get variant Creutzfeldt-Jakob disease. So the little v, so notice that Creutzfeldt-Jakob is also a genetic disease, but the variant of it is because you ate some meat contaminated with that prion. So this was a big issue. I think it was in the 90s where people were getting prion disease from downer cows. So cows have to be healthy to enter into the human food supply. So if a cow is looking sick or acting weird, it's not going to be part of the human food supply. So I think there was some issue with that where downer cows were getting into the food supply and they had the mad cow disease. You can also see there's a couple more of transmissible diseases. There's scrapie, sheep and goats, and then kuru is a laughing sickness. This is really interesting. Just one little 30 seconds on kuru. It's from a Papua New Guinea tribe or group of people who actually practiced cannibalism. I think they stopped at this point, but one thing that they would do is eat the brains of the dead. So if someone had this laughing sickness, this neurological disorder of prion, and then you ate the brain of the person who died, right? Absolutely, you're going to get this prion disease. So that is another transmissible form of this disease caused by prion. And so you can see here, this is a schematic of our endogenous. What endogenous means is that it exists in our body. So PRP stands for the protein, prion protein. And then when it changes shape, like literally all that happens is that it becomes misfolded in some way. So this is the misfolded form. And then this misfolded form can actually interact with the normal form and cause the normal form to also become misshapen. So if you have a lot of these around, they're eventually going to turn all of your normal prions into this infectious, weird type of prion that tends to clump and accumulate. And then this is an image of some nervous tissue, brain tissue that has those areas that are clear, right? These areas are holes, and those holes are caused by that prion. So this is a normal brain. You can see that the CJD brain, there are lots of areas that don't contain a lot of tissue anymore, a lot of areas with big gaps, and then the normal brain tissue in the CJD, where you have those areas of nothing, right? No cells, because it's a spongiform. So the brain of a CJD patient is full of sponge-like lesions. Okay, one human disease caused by prion is... it's going to be Creutzfeldt-Jakob. All right, and to end this one, we have a viroid. So viroids are little circular pieces of RNA, and they usually just infect potatoes or plants. So a viroid is typically spread with knives that you use to cut healthy potatoes, and then you can get the virus on the knife. But viroids do not bother humans. It is another infectious agent, though, that is non-cellular, and it does cause disease in these potatoes. And that's it.